All right, good afternoon, everyone. And thank you for joining us for our final session of the Extension Master Gardener IDs and Diagnoses series. We've had such a wonderful turnout and engagement over these past few sessions, and I really appreciate all of your time and attention for all of you who've joined for one or more sessions from the past week. This session is being recorded and is going to be posted to our webinar archives by the end of this week. And you can also find all other session recordings from the past weeks on that page as well. Please use the chat box to ask any questions you have for our speakers, and we will get to as many of those as we can at the end of the session. So without any further ado, I'd like to welcome our speakers this afternoon, Elizabeth Bush and Mary Ann Hansen of the Virginia Tech Plant Disease Clinic. Welcome to you both, and thank you so much for joining us this afternoon.
Thanks.
I guess should I get started, Kathleen?
Yeah, go ahead, Elizabeth.
I'm Elizabeth Bush, and it's a great opportunity to see so many master gardeners. I even see some of you in your house, which is amazing. And it's great that we can reach out to you this way with some information that we hope you'll find helpful. I'm going to start off with the first presentation, and I'm going to focus on digital diagnosis and also some resources that I think you might find useful. And Mary Ann Hansen, who's the other diagnostician in the plant clinic, will follow up with a presentation focusing on physical sample submission. And then we're going to save about 15 minutes for questions at the end. So we've learned a lot in this past year where we've done so much intensive digital diagnosis. As some of you may know, we've only been accepting physical samples from commercial growers since about April of last year due to the pandemic. But we found that we could make accurate diagnosis from digital images more frequently than we expected. We've been very pleased with that process and surprised. We do need good digital images, and good digital images can also provide important context for diagnosis even when we need a physical sample. So that's always been useful. But we also realize that clients really need instructions on taking proper images for diagnosis. One thing is the images need to be focused, which is pretty obvious, but it wasn't always the case that we would get focused images. And we need good close-ups of the symptoms, but that's not all. If it's a foliar problem, we really need both leaf surfaces. And a picture of the whole plant provides a lot of information to us because we can see the pattern of the problem on the whole plant, rather than just the affected part of the plant. And if it's a tree, we like to see the base of the trunk as well. One thing that people often forget about is to include a picture of the plant in the landscape or whatever other setting it's in, like a greenhouse. That allows us to see the pattern of the problem in the landscape and can also provide a lot of clues if the problem's abiotic particularly. What we do know is that digital diagnosis is going to be a service that we're going to offer going forward. And with that in mind, we have a new feature that we've successfully beta tested with a group of agents and made some modifications to. And the feature is that it's a direct uploading feature from the offices into our Clinic diagnostic database. So I'm not going to go into a lot of detail on how that works since we've released detailed instructions on that process to extension agents and offices. There is a sign-up process, and if your office has a Master Gardener help desk that handles diagnostic samples, then your supervising agent or staff can talk about that with your group. But basically, what happens is the images for a digital sample are uploaded by the agent or whomever in the office, and they also upload a completed diagnostic form. That we see it on our end, and we can take a look at the pictures and see if it's we're going to be able to diagnose it, if it's a sufficient sample. But if it's not, we can decline that sample or reject it. And in that case, what happens is you would get an email, whoever uploaded it, that says, we're not able to process your sample. And that could be because maybe the images are poor quality, or maybe the range of images that we want. For example, the whole plant and the landscape were not included. Or maybe we need a physical specimen. Maybe it looks like a root problem and we really need the roots to diagnose that. So whatever the issue is, an email would come to you and tell you exactly what the problem is. And who can directly upload to pClinic? Well, the VC agents and staff can do this. There is a sign-up process, as I said, and a Master Gardener help desk can if requested by the supervising agent and staff. And we do expect the supervising agent or staff to provide oversight and training to the Master Gardener help desk in that case. There are some benefits to directly uploading samples to the pClinic database. A really great one is that there's an electronic record of all samples from your office that have been directly uploaded. So there's a historic record that can be accessed by the agent or the county upload group or the Master Gardener help desk. They can log in and they can see all the samples that have been submitted electronically. They can look at the images that were associated with that sample. They can look at the reports that we sent out on that sample. So that record is there and accessible at any time. The other thing is, if the sample is insufficient, you get a quick notification and we can say exactly what we need. If it's better images or a range of images, or if we need a physical sample, we can send that out. And we can also tell you what kind of physical sample is needed. You know, if we need the whole plant or if we just need a root sample, maybe even information on how to package that sample. One of the things that clients have really liked and agents have liked is that there's a faster report on samples that can be diagnosed from digital images. And of course, there's no need to mail a sample in. So if we can accurately diagnose something from digital images, it's a real time saver. And another thing, when we are accepting physical samples, again, from non-commercial growers, it's great to have, well, people always have sent images into our lab for some samples, but they'll just send an email and they'll attach the images and they'll say, well, the sample's coming in the mail. And then we kind of have to remember to associate that with the sample when it comes in. So this streamlines everything on our end as well. I mentioned, well, I don't think I did mention this, but I did mention the need that we realize that clients really need to be educated about what kind of digital sample we need. And so we made this 2-page PDF that would be great for you to use, share with your clients or use for yourself to know what kind of a digital submission is good. And it's a printable, downloadable 2-page PDF. So basically it talks about, you know, the sizes of the image. The images can't be too small. We have to zoom in, but they also can't be too big. What type of images, but most importantly, it shows the range of images with some actual examples. For example, you'll see the boxwood down here on the first page, and you can see there's a good close-up of the boxwood, picture of the whole plant, and then a really important picture of the problem in the whole landscape. And there are also some examples of well-focused close-up images. So I was going to share some good digital submissions that have come into our lab, and also some that aren't so good. So this was a Mayapple sample, and this is again the front, the top, and the bottom of the leaf surface. And this is a good sample as well because it's taken on a neutral background. Sometimes people take pictures on a really busy background, and it's hard to to see the features. It's also a good picture because it doesn't have a lot of shadows on it. So sometimes if you take a picture on a bright sunny day, you can get a lot of shadows. And so this was distinctive enough that I could diagnose rust on Mayapple from this image. This was a U sample that came in, and unfortunately, this was the only image they sent. So they did not send the range of images that we requested, but it is a very good image in that, you know, it's well focused, and I can see the pattern of the problem, at least on this section of the U, is very uniform, and only the new growth is affected. But again, they didn't submit the range of images that we would really like to have. But they did complete the diagnostic form well. And on the diagnostic form, they had listed that all 10 of the plants were affected. And that's another clue to me that it's likely an abiotic type of problem rather than a disease problem. And this was in June, early June, and I knew that we had had some really severe late freezes, both in April and May. And I looked up the weather history for this county, and that supported my hypothesis of freeze injury. So I was able to diagnose this, even though they didn't send the range of images, but that would have been helpful. But it was important they did complete the form very well. That's always important information for us. Here's a few more examples of good images. So this is a peach. And this was taken on the tree. We've had a lot of good images that people have taken right on trees of foliage and things, but notice that there aren't shadows on the peach. I could see the symptoms really well, and so I could confidently diagnose this pretty common problem on peach as brown rot. Here's another image of some ornamental pear that has a disease called cedar quince rust that's very characteristic, and so an easy disease to diagnose from images. The good thing about this image is they took it on a neutral background. We could see the features well, and also there aren't shadows again. So taking pictures on a cloudy day or early in the morning before it's very sunny is a good practice. And this again is another image that was taken on a neutral background and without a lot of shadows, and I could see the characteristic symptoms of oak leaf blister on oak leaves. So it's been really fantastic that we could diagnose a lot of these things digitally. And with smartphones, you can take very good images, so it's easy for people. This is an example of a really good digital submission that shows the range of images that we want. It shows an overall image of the affected plant in the landscape, the tree in this case, and it shows close-up images of the leaves that are well focused. There aren't too many shadows, and they took a picture of the base of the trunk, which we wanted. So this is really great, especially for trees, which can be difficult for our lab. I think the images really, really help, especially with trees. So here's some examples of some not so good images. This is just a snip I took of what we see on our end when something's uploaded into the database. So the PDF is the completed diagnostic form. And there were actually 10 images that were very similar to this. I couldn't snip them all and put them on the screen, but sending 10 bad images is not any better than sending one bad image. So this was an insufficient sample. They didn't submit the range of images, and basically it's just some dried up leaves on a branch without any distinctive characteristics. This was another sample that came in. In this case, they did take good pictures of the affected plant part, but basically they sent three images that are basically the same thing, and they're all kind of close-up images. They didn't send an image of the whole plant, and they didn't send an image of the plant in the landscape. The good thing about it was that the images were large enough that I could zoom in. They had sent this as a boxwood plant, but at least I could see that it actually was a Japanese holly. So in that case, I suspected that it was probably a certain disease that we often see on black holly, and I could at least kind of suspect that, but it would have been better to have the whole range of images. And in this case, if we were submitting physical samples, I would request certainly a root sample. So now I'm going to switch gears a little bit and talk about some resources that I think might be helpful to you that are on the Plant Clinic website. So if you want to find that website, just Google Plant Disease Clinic at Virginia Tech and it should pop up. We have a number of tabs here, and I'm going to go through some of these. So I want to point out just that the current Plant Clinic sample submission policy is posted on our website under submitting samples. So right now we're still under our COVID policy, and that's posted here. But if it changes, that will be updated. And I want to point out that that guidance for submitting a good digital submission is also posted here. So you could download that 2-page PDF here or share it with clients or whatever. A lot of master gardeners often ask, what are the common disease problems that we diagnose? And I always say the pest management guides are a great starting place. And on our website, you can link to the pest management guides. To my knowledge, I think we're the only place that has all these guides together in one place, and I find it handy. I use it all the time. So the most relevant one is the Home Grounds and Animals Pest Management Guide for your group. But you might find some interesting things in the other ones as well. That's, if you don't know already, a PDF. That's accessible, and it's also broken up into sections. So the sections are right here from this table of contents. So you can click on these sections on the web and open one up at a time. But I like to tell master gardeners it's good to become familiar with the Home Grounds and Animals Pest Management Guide. It's a little, it takes some time to do that. So it's worthwhile to try and spend some time at it. So I'm going to show you the different sections that are relevant to what I'm talking about, which is disease problems. So the home vegetable section, there's a home fruit section, home ornamentals, which is broken up into ornamental diseases and landscape trees. And then there's a short lawn section. And all of these sections are organized somewhat differently. And that's why it's a little hard to get a handle on, but there are various reasons for that. So for example, the home ornamental section has woody plants, and it also has herbaceous plants. And at the beginning of those sections are, here's an example of the herbaceous plants, which are listed by their name. Whoops, sorry, went too far. So if you look at this list, for example, snapdragon, it shows the common problems on there: Cercospora leaf spot, downy mildew, phytophthora root rot, etc. And then after this list, you can find the fungicide recommendations for those different diseases. And that's how the home ornamental section is organized. The landscape trees is organized a little differently. We have the common tree names listed. For example, ash, beech, birch, black gum. And then after that are the common diseases, the most common diseases we find, like anthracnose and rust on ash, and black gum, felt fungus, canker, various cankers, cryptocline leaf spot. And beside that are the chemical controls, and we also have cultural controls included, and any special precautions or anything. The home fruit section is interesting. There's a lot of things buried in that section that you might overlook if you jump straight to the spray recommendations, the fungicide spray recommendations. For example, here you can see the common diseases. Some of the most common diseases that we see on apple in Virginia are powdery mildew and cedar apple rust, quince rust, and fire blight. And a list of cultivars recommended for planting in Virginia that are resistant to those diseases or have some resistance to one or so of those diseases. So that's just another thing that there's to point out. There's more information in that section than you might be aware of if you don't take a look at it. Another place to see common problems diagnosed are the Plant Disease Clinic annual reports, which Mary Ann puts together every year. And those are organized by the plant type, like a tree, the ornamental, woody ornamentals or field crops. And within those are the common names. So here you can see, I pulled up dogwood, and you can see powdery mildew is diagnosed frequently, and spot anthracnose follows that. So that can kind of give you an idea of some common problems. This is something I think you all would enjoy looking at and be more excited about probably, which is the Plant Problem Image Gallery, which we have with support of IT help from CALS, our college, recently resurrected back in 2019, and we've been adding problems to. And this is also linked on our website. And basically, it has drop downs. You can drop down by plant type, plant common name, or plant scientific name. Here I've picked dogwood, and you can see below this populates with all the problems that we have in the image gallery on dogwood. And you could also search by pathogen type, like if you want to look at all fungal diseases or the plant problem name. But if you clicked on, say, septorial leaf spot on this list, this is what would open up. And you would see a picture of the disease on dogwood. And below this, you would see a line of images below this. There's actually more than one image in here, but I couldn't snip it on the screen. So this one also has a description of the problem. And usually that will also have a link maybe to the home management guide, the pest management guide. So here's another example where you can see there's more images with the sample. So this is Augustaceae, a bacterial leaf spot. And if you clicked on any of these images, you would be able to enlarge them, or you can view them in a slideshow. But I'll point out that not all the problems have a description yet. And that's something Mary Ann and I need to do, and it just takes time. We've had a student uploading a lot of these images, but she can't do that part. But I want to point out to all of you that all of these images have a Creative Commons license, which means that you're free to use them in any way for non-commercial purposes. So that was the whole point of this database, really, is for agents to have access to images to use in their educational outreach, and also master gardeners. In fact, I had a master gardener from, I think, Lynchburg, wanting to know about images, and I think she used images from this resource. So here's actually foliar nematodes on anemone. That's another example. And this past year in 2020, we've basically doubled the size of the, about doubled the number of plant problems and the number of images in the database. And our goal is to continue to add problems and images because basically we have a whole diagnostic career worth of images. So I think this will be a good resource going forward. So that concludes my portion of the presentation. So I'm going to stop sharing my screen and let Mary Ann share. And I thank you for joining today.
Can you guys hear me?
Yep.
Yeah,
Okay.
you sounded good.
I'm gonna, I also want to try out these earphones and see if does that make the audio a little better?
You're sounding good.
That sounds good. I'll leave those in. Okay, great. Thanks for leaving me a little extra time, Elizabeth. We wanted to divide this talk into some of the new resources in the Plant Disease Clinic, and then spend the last half of the talk talking about how to prepare samples to send to the Plant Disease Clinic. And we know that can't really submit samples right now because of our COVID policy, but we hope that that situation will change hopefully in the coming year. And we think this is a pretty important subject to cover because you won't get a good diagnosis unless we get a good sample. And so we wanted to spend a little time talking to you about not only how to submit good digital images for now, but also how to submit good physical samples in the future. And, you know, it's our student helpers who open all these boxes that you guys send and who log all of that information in. And so I'm just gonna change my view here. So we want to keep them happy. And we'll see through this presentation that some samples will keep them happy and some samples will not. So let's get started. You know, the first thing I think that many of you experience and think about when you send a sample to the clinic is that sometimes you get a report back that says insufficient sample. And Elizabeth addressed that a little bit with some insufficient, showing you some insufficient photos. But we also get a lot of insufficient samples in the clinic. And this is, you know, represents basically a waste of your time and a waste of our time. I know it's really frustrating to get a report back that says insufficient sample because you've spent time packaging that sample, getting the people to fill out the form, sending it off, you know, waiting for the days in the mail and the time in the lab, and then you get back a report that basically puts you at ground zero again. So I want to address that a little bit today to give you some ideas about how to avoid that. I think that these samples pictured here are probably pretty obvious to you why these are insufficient. These were samples where, you know, people waited till the plant had totally died before they gathered it up and sent it in. And what happens on dead plant material is that secondary decay fungi and bacteria invade them. And so then it can be hard to find the original pathogen or maybe even insect or whatever caused that problem in the first place. So this is one type of reason why you might get an insufficient sample report back. And, you know, I know a lot of times these people are coming to you, they've already collected their sample. You hate to tell them that it can't be, you know, diagnosed. And so you send it in. But I think that you need to learn to educate people about why these samples are not going to be useful, and then maybe they'll be more willing to go home and gather another sample. Another way that a sample could be insufficient is if you send the wrong plant part for the plant part that's affected. So maybe you've got a root disease and you're sending in a leaf or a stem sample. We cannot figure out that it's a root disease unless we get roots. So I'll address that a little more as we talk about some of the sample types that you might send. First, I'm going to talk about some general tips for collecting all plant samples, and then we'll talk about some more specific types of plant samples. So one thing that applies to everything is that you want to collect samples that are in the early stages of disease. You know, if they look like those samples on that first slide, they're just not going to be useful for diagnosis. So here's a pretty good example of a lima bean sample that came in. You can see a whole range of symptoms from early stages to late stages, but you can still make out those individual leaf spots. You can even see the fungus sporulating on the edge of those spots. And so that's the kind of thing where we're going to be able to find the pathogen that caused that problem. Here's a different sample that is not quite as fresh. We couldn't really figure this out. This came in a jar and it was called mustard greens. And we weren't sure if they wanted us to taste these mustard greens or if they had had some kind of disease and completely rotted. In any case, this was not adequate for any kind of diagnosis. And it's just, you know, too far gone. And this is the kind of expression that you're going to see on the faces of our student workers when we get that kind of sample. So you want to try and make sure you submit a fresh sample. Here's another good example of a sample in the early stages of disease. You know, luckily in this case, we got a whole plant submitted, and you can see a close-up of the symptoms on the leaves with those characteristic ring spots suggesting a viral disease. If that whole leaf had turned brown, you know, we wouldn't be able to see those symptoms. So this is a good example of a plant that we were happy to get. And we're blowing you kisses, thanking you for those. Another point that's generally true for all plant samples is that you want to collect a sample that will show the junction of the healthy and the affected tissue. So for example, on this stem sample here, you see a canker. That's the brown area on the stem, and then you see a margin with the green tissue. And that's the place where, if there's a pathogen, that's where it's most likely going to be located. It's going to be where we can culture that pathogen from. So pathogens will typically move from the dead tissue into the healthy tissue. And, you know, meanwhile, the decay organisms are invading that dead tissue. So we want that junction of the healthy and the affected tissue. And even if it's on the inside of the stem, so here's a case where you can see that junction on the inside of the stem after you cut away the bark. We don't really want you to cut away the bark, but you can recognize that junction by looking at the plant. You know, if it's got one dead stem attached to a healthy stem, we want to get a sample that includes that junction. Don't cut a dead stem and send it separately from a healthy stem, because you're going to miss that junction, which is where we need to culture from if we need to culture. So look for those junctions, and please know that bark chips are not useful for diagnosis. We get a fair number of bark chip samples, and basically you're not going to see that junction on a bark chip, and they are not useful for diagnosis. It's sort of like, you know, clipping your toenails and sending them to your doctor to have him or her diagnose a heart disease. So please, no bark chips. Okay, another thing that would seem like maybe it's a no-brainer, but if you don't have a lot of experience sending samples, you might not know this. You don't want to wrap samples in wet paper towels. As the samples are in the mail for several days, if they're in a moist environment, they're going to decay even more rapidly than they normally would. As many of you probably know, most fungi and bacteria are going to be much more active in a moist environment. So do not add a wet paper towel into your plastic bag that you put your sample in, and don't wrap your sample in it either. And please do not wrap the sample in the diagnostic form. That's not what the form is for. We literally got this sample years ago wrapped in our old carbon copy form, and there you can see the expression on the student workers' faces. So if you collect a sample and the leaves are still wet, maybe there's still dew on them, you've collected in the morning, you can blot the leaves dry with a paper towel, or you can simply add a dry paper towel to the bag, but just don't add a wet paper towel to the bag, please. So as I intimated in the last slide, most samples can be placed in a plastic bag, and that will keep them, you know, moist, but not too moist, as long as you're not adding wet paper towels. So that's fine. The only exception would be mushrooms. So we do get some mushrooms for identification, and those will rot very rapidly in a plastic bag. And I'll talk more about that a little bit later, but you want to use paper bags for mushrooms. But some people will put plant samples in paper bags, and they just completely dry out, and we can't often see the symptoms that we're looking for by the time we get the sample. And then you want to be sure to include a completed diagnostic form with your sample. And that form, as was mentioned earlier, is available on the website. So you can go to the sample submission policy document that we have there now, and you'll find a link to the form. And you want to submit that form with the plant sample. Here's a very nice example of a sample of some soybeans that was submitted with the form, and they took the time to put the form separately in a Ziploc bag so it didn't get, you know, a lot of soil or plant tissue on it. So our student workers were happy campers when they saw that one. When they saw this one, not so much. Here, the person at least put the form in an envelope, but then they threw the envelope in the bag with the soil and the plants all jumbled together. And then, in that case, typically the form will come out of the bag totally wet and dripping. So don't put the form right down there in the bag with the plant sample. And then mail early in the week. If you're mailing on a Friday, then that sample is going to sit in the mail in the plastic bag over the weekend. If it's a holiday weekend, it's going to be even longer. And by the time we get it, it's probably going to be pretty rotted. And you can see that the look on the student worker's face is just exactly like this look I showed you before. So try to mail early in the week. If you get a sample delivered to you on a Friday, then find a refrigerator and put it in there until Monday and then ship it on Monday. Got to keep those student workers happy. As Elizabeth mentioned, we have really discovered that digital images are so useful. And right now, that's basically all you can submit. But even when we go back to accepting physical samples, it is useful to get those digital images as well, especially for things like turf, where it's hard to kind of see the big picture unless we get some good pictures, and for trees, where we typically are just getting part of the tree and we can't see, what the symptoms look like on the whole tree. So submitting those digital images, even if we can't diagnose the problem just from the digital images, they're going to be a useful additional tool to the sample we get, since that doesn't really give us the whole picture usually. Okay, I want to talk in a little bit more detail about the types of plant samples that you might send us. And so we'll start out with leaf diseases or leaf problems. And, it may be a little bit hard to tell whether something is originating on the leaves or not. But when you might suspect a leaf disease is when you see scattered spots on the leaves, or in this case, you're seeing kind of scattered irregular blisters. This is a disease called leaf blister on elm. Or you might see scattered galls. So those sorts of things, clearly, whatever's causing it is attacking the leaves directly. And so in that case, we don't need a root sample or a soil sample. We just need the leaves, but we want them still attached to the stems because they'll remain fresher that way. So this is a good sample. Of course, we'd want a few more stems. You want to give us, a range of symptoms. So several stems is good with the leaves still attached. And then just put them in a sealed plastic bag and mail them in a padded envelope or padded box. Here's an example of a leaf sample that we did not like. These are just some dried out boxwood leaves and scattered twigs. And this is not very helpful. These desiccated leaves, if that's all we get and no pictures or anything, it's going to be really hard to tell what caused that. The leaves could have dried out because of drought or root rot or almost anything, and those leaves are not going to be very useful. Now, I will say that now that we have boxwood blight in Virginia, one of the symptoms is severe leaf drop, and it can be helpful to pick up some of those leaves and mail with the sample. But in general, you don't want to be sending leaves that have dropped from the plant. You want to be sending leaves still attached to the plant. And in the case of boxwood blight, we would still need the stems with that sample. For stem diseases, the main thing to remember is that you want to include that junction of healthy and affected tissue. So wherever you can find an edge, if you can see lesions, or maybe you can see the edge of a canker. Canker is just dead bark, and cankers can look different. They can look like that picture I showed you before, where you've got brown up against green, or maybe there's just swollen tissue or sunken tissue. You want to include the edge of that canker, and then send us multiple stems in a plastic bag in a padded envelope or box. For root or crown diseases, and with crowns, we're talking about the base of the main stem or the base of the plant. Whenever possible, we know it's not always possible, we would like for you to send a whole plant. So that way we can look at the whole root system and decide, if we need to culture from the roots, then we can find the roots that we need to culture from. So whole plants are always preferable, but we know that's not always possible, certainly for, woody plants like shrubs and trees. But in cases where it is possible, that is really helpful. And then you want to leave the soil on the roots, and just with, whatever level of moisture it had, don't add water. It will help us to see, what the condition of the soil was. That can be important information for the diagnosis as well. And, if the soil is relatively moist, then the roots will remain moist as well. And then you want to place that root system in a plastic bag and tie that off around the base of the stem, leaving the aerial parts of the plant outside the plastic bag. That way the soil won't, fall up on the leaves and make a mess of the sample in the mail. And here I pictured a plant in a pot, but you don't need to include the pot. You can take the pot away and just put the root ball in a plastic bag and tie it off around the base of the stem. Now, in cases where you cannot send a whole plant, it is still possible to diagnose a root disease from a handful of the fibrous roots. So if the people are only willing to send a root sample and not the whole plant, then encourage them to take several large handfuls of the fibrous roots from different parts of the root zone and leave those in the soil and package that up in a plastic bag. And sometimes people will go out to dig up roots from their landscape, and they dig up soil and they think there's roots in there, but when we get it, we discover there are no roots in the soil. So if you're doing that, then make sure there are actually roots in the soil. Or if somebody's bringing you a sample, you might need to send them home again and say, you need to collect some roots. Again, with root diseases, if we don't have the roots, we typically can't diagnose the problem. And then once you've, wrapped up that root system in a plastic bag and tied it off around the base of the stem, you can package those plants in a box with some padding and mail it off. This is a good sample. They had 2 plants clearly labeled. Each one had the root system in a plastic bag tied off around the base of the stem. Nice sample. As opposed to this sample, which was a jumble of some boxwood twigs and 1 root, not in soil, not very helpful. That root's going to dry out in the mail and not be very useful for culturing from. So this is an example of a sample that we were not happy with. Fruit diseases, basically you want to package fruit in the early stages of disease in a padded box, so it's not jumbling around. Fruit will tend to rot very quickly when it's bruised, and so you don't want those fruit samples to be knocking around in the box on the way here. And here are some good examples of acceptable fruit samples. The apple has some scattered spots. It's not totally rotted yet, and the grapevine there has both dead and healthy grapes, so it's probably in a stage of disease where we can still find the pathogen. So those are good samples. This is the kind of thing where, once the grapes are all shriveled and dead, they're probably not going to be very useful for diagnosis. And we have, especially with tomatoes, we sometimes end up pouring those out of the bag. Turf is kind of a special case, and turf diseases can be kind of difficult to diagnose, partly because turf is composed of a lot of different individual plants all growing together, and there can be several different species of turf grasses in that turf, each of which gets its own disease. And so we kind of have to figure out what species of turf are in there, and it can be a little tricky. So one of the good things about turf diseases is that a lot of them occur in patches, so dead spots scattered in the turf. And so what you want to do is sample from the edge of that patch. And you want to include, again, the junction of healthy and affected turf. So it's the same thing for every different kind of plant sample. And I think you can clearly see it on this turf sample that was submitted here. they did a nice job of collecting a plug of turf that was about 6 inches in diameter. And it was right at the edge of that patch, and it showed the dead and the green tissue. And it was sent in the mail, and in the plastic bag in the mail, the fungus that was causing the problem fluffed up, and you can see where it is. It's right at the edge. You know, like I was saying, it's moving from the dead to the healthy tissue. So that's a very clear example of why we need that junction of healthy and affected tissue. And then with turf, you want to make sure that you're also getting the roots. So dig your sample deep enough to include the root system of those plants, because it could be a root disease. And again, without the roots, we're not going to be able to tell. And then you can just wrap your plugs in a plastic bag in a padded box for mailing. So one question is, you know, when do you need to send roots and soil? Because roots and soil are kind of a pain to get. It's a lot easier to send a leaf or a stem sample. And so I want to give you a few tips on you know, how to decide when you need to send a root sample. Basically, any injury to the roots is going to cause symptoms in the above ground parts of the plant. So even though you can't see the roots, you can probably surmise that you might have a root problem from some of these above ground symptoms. So if you have a lot of dieback, that's not just scattered individual branches, but, you know, whole sections of a plant, or maybe the whole plant, or wilting of the whole plant, stunting, general chlorosis, which just means yellowing. All of these symptoms could be due to impairment of the root system, and then the roots are not transmitting water and nutrients effectively to the leaves. So if you see any of those samples, then you want to be thinking, I may need to send a root sample. And here's an example of a dogwood tree that we actually got the whole tree in the clinic. It was a local person that had this tree, and the whole tree had wilted, and they brought this tree in. And here's an example of a case where if you would have taken a separate fibrous root sample as opposed to the whole tree, then we might not have been able to diagnose the problem. I think you can look at that picture and you're probably asking yourself, how could that root system be so small for that tree? That's what I was asking myself. And, you know, that wasn't a very big root system to support that tree. And what happened here was something that is very, very frequent, which is that this tree was planted too deeply. So when we got it in, it had soil all around the roots. And I have learned to take these trees out back and to chip away the soil and to mark the original soil line before I do. And so this was where the original soil line was. That tree was planted about 8 inches too deeply. And what happens is that the roots are too deep in the soil. They're not getting enough oxygen. They can't function in transporting water and nutrients. Now, the grower said that this tree had wilted overnight. So to them, it seemed like a sudden problem. But you know what probably happened here was that root system was able to support that tree for a while, and then once it had too much foliage, that root system couldn't support that canopy, and the tree just collapsed. So it looked like a sudden problem, but in fact that had been a problem that had been there for a long time. And that's the kind of thing that we're not going to be able to diagnose if you send us just a sample of the fibrous roots. We can tell you, you know, we can send you a report that says no pathogens were found after we culture from those roots, but we're not going to be able to tell you what the specific abiotic problem is the way we might be able to if we get a whole plant. And again, with a sample like this, we got the stump of a pine tree and it had girdling roots. Another very common problem where plants have been grown too long in a container and then they're just planted in the ground right from the container, and they never grow out into the soil in a way that can support good growth of that tree. So that's another case where if you dug up a fibrous root sample, you know, we could tell you no pathogens found, but you know, we wouldn't be able to tell you much more without seeing that whole root system. Here's another symptom of a root disease, dieback in Japanese holly. And here's a case where we could diagnose this with a root sample. So if you sent us a handful of fibrous roots, we would be able to culture from those roots and tell you what the disease was, in this case, a common disease called black root rot. And another thing to remember is that it's always good by default, if you are sending a root sample, to make sure that you're sending at least a pint of soil. So for certain plants like boxwood, for example, we will automatically process that soil for nematodes. So on plants where nematodes are a common problem, we will process the soil for nematodes. And nematodes, we extract them from the soil, so we don't culture them. We get them out of the soil. So if we don't have the soil, we're not going to be able to do the nematode assay. But you know, when you're looking at this soil sample that came here, I want you to realize that this is not a pint of soil. So we need a whole pint. And those of you who cook kind of have an idea of how much a pint of soil is. We're not going to be able to do much with a few little dribbles of soil like this. So make sure you include a pint. And basically for herbaceous plants, we would really like for you to encourage people to send a whole plant, because it's going to be much easier to diagnose a root problem on a herbaceous plant from a whole plant. And here you see this plant was nicely packaged. Here they wrapped the root system in Glad wrap and left the leaves, outside the Glad wrap and sent it in a padded box. And that was a happy sample for our student workers. Occasionally we get samples for plant identification, and while that's not the main goal of our lab, we do some plant identification, and we work with other people on campus like Jordan Metzger in the herbarium and Alex Namira, horticulturist, who are good at identifying plants. So we will do some plant identification as long as it's not weeds. So the weed samples need to go to the weed identification lab. But if you are sending us a plant for identification, then you want to send the plant sample at a time of year when you can collect some of the reproductive parts like the flowers or the fruit or the seeds, because those are often critical to the identification. And you also want to make sure to leave the leaves still attached to the stems, because often the leaf arrangement is involved in plant identification as well. Mushrooms, as I mentioned earlier, are kind of a special case where you do not want to mail them in a plastic bag. They will just disintegrate in the plastic bag. So mushrooms you can put in paper bags, but plant samples you want to put in plastic bags. And basically mushrooms, if they dry out in the plastic bag, they can be rehydrated and we can still identify them in some cases. But plant samples, once they dry out in the paper bag, if you would send them in a paper bag, they would dry out and we would not be able to rehydrate them. So mushrooms are kind of a special case. And I just want to mention too, for mushrooms, basically our expertise is in identifying plant pathogens. A lot of mushrooms are not plant pathogens, and we have lost our mushroom expert in the biology department. So we do the best we can, but we do not claim to be mushroom experts, and we do not make any declarations of edibility for liability reasons. So just be aware of that. Okay, samples for insect or weed identification should go to those respective labs. So if you know that your plant has an arthropod problem, either an insect or a mite problem, then you want to send those samples directly to the insect ID lab. In a case where you're uncertain about whether it's an insect or disease, then go ahead and send that sample to our lab with our form, because we require a lot more information about our samples. So then if it turns out it's a disease, we'll have that information. And then, if we decide that it's an insect problem, we will forward that sample to the insect ID lab. We have a very close working relationship with Eric Day in that lab, and so that is no problem. Weed samples should go directly to the weed identification lab. So we only do plant IDs that are not weeds, and you can find information about submitting samples to those labs on those respective websites. So I want to go over a little bit in detail the diagnostic form, which is available to you on the web. And I think those links have been posted in the chat, I believe. And in any case, you can go to the plant clinic website and you can get to it there. And that form can either be completed electronically or you can print it out and fill it out by hand. But the important thing is that you want to complete all the relevant sections of the form for your sample. And you want to remember that there's 2 pages to that form. A lot of times people only fill out the front page. But all of the information that we're asking for is very important to the diagnosis. You may not have all of it, but you want to fill out as much of it as you can, because that background information is often just as important as the sample itself for making an accurate diagnosis. So let me just go through and highlight a few sections of the form. And, obviously you want to put what the plant is. Maybe you don't know, but hopefully you do. And that is helpful. If we see that you've identified it as the wrong plant, we will correct that. But definitely put the plant name in there. You want to describe the symptoms that you're seeing to us in part 3 of that form. And by describing the symptoms, I don't mean to write, please diagnose and recommend control. That's what we're there for. And we're going to do that. What we need for you to be our eyes. So, you want to write down the symptoms that you're concerned about, but also any more information that you can tell us about things that we can't see, like symptoms on the whole plant, if you're just submitting a plant part, or symptoms in the whole planting, which we can't see unless you send us pictures. So that's the type of information that we want there that is critical to the diagnosis. You want to make sure to tell us when those symptoms were first noticed down in section 5. And fill out all the other information as well, but some of it is pretty straightforward. But the reason we need to know when the symptoms appeared is that we might be able to associate that with either a time of year when a certain pathogen occurs, or maybe with some weather that has happened just prior to symptom appearance, or maybe to a chemical application. So let us know when the symptoms were first noticed. If you noticed them 3 years ago, then chances are we're not going to be able to diagnose the problem at this point. But be as specific as you can. Weather is important. Certain weather conditions can favor certain pathogens. And of course, we can have abiotic problems like the frost injury that Elizabeth showed in one of those pictures that she showed. So you want to be telling us what the weather conditions were just prior to symptom development. And then on page 2 of the form, section 9, that section asks you for any information about chemicals that have been applied, including rates and timing. And, you may not have all this information, but if you do, put it in there. That can be really helpful. If your sample has chemical injury, which is increasingly common these days, then we need to associate the specific symptom with the type of chemical that was applied, because different chemicals will cause different types of symptoms. So the more information we have about what was applied, the more we'll be able to rule out or rule in chemical injury. And then very importantly, section 10, the woody plants section. This is really critical for woody plants. So, woody plants a lot of times have been in the landscape for a long time, and the symptoms on them may relate to something that happened a lot earlier. And so we need to know, how long has that plant been in the landscape? If we don't have the whole plant, we need to get a picture of how many dead limbs there are and that sort of thing. Any construction injury around the roots, all of this can be relevant. So be sure to fill that out for any woody plant. And so the bottom line is that for accurate diagnosis, we need a good sample. And by good sample, I mean following some of these tips that I've given you today. And we need a completed form. So one thing that you can really do as a master gardener, is educate people who are bringing you samples about why it's important to fill out that form, why it's important to give you a good sample. And of course, why it's important, the ultimate reason why it's important is that you are keeping our student workers happy. So that's something we want to do at all costs. So you think about their face next time you send us a sample, and hopefully you're putting a smile on their face with your sample and form. And that's all I had. So at this point, Elizabeth and I can both take questions, and I'll stop sharing my screen. Oops.
Wonderful. Thank you both so much for such detailed and important information to share with our Master Gardeners. Those are two very helpful presentations. So thank you. There have been a few questions that have come in. So the first one is, will there be any opportunities post-COVID for Master Gardeners to visit the lab and see some hands-on diagnosis?
I'll take that one, Elizabeth, because I actually wrote an answer to that in the chat while Elizabeth was speaking. And, you know, we have typically done a lab tour for the Master Gardener College, and we'll probably continue to do that. It's a lot more involved to set up a hands-on workshop, although we have occasionally done that for Master Gardener College as well. So it's possible we would do that in the future. And then I will just mention that, you know, some counties have been going over to more of a model for Master Gardener training, where they have the trainees, you know, do some work ahead of time, and then they use the training session to do kind of a hands-on lab. And so I've done some portable labs with counties in that way, and that's something that, you know, you may want to look into for your county.
Great, thank you. And the next Master Gardener College on campus in Blacksburg will be in 2022, since our office is hosting the International Master Gardener Conference this year. So we plan to see you back in June of 22.
Great.
The next question is it all right to fold up a larger plant sending a sample in the lab?
Yeah, I think I answered that in the web, but what I said was it depends. You know, if it's gonna ruin the plant, you know, if different plants are different, so I can't really give a blanket answer on that. But if it's gonna break up the plant and cause it to be damaged and wilt, and we're not gonna be able to see the symptoms, then that wouldn't be a good approach. But I offered an example of a large tomato plant. We usually need whole plants with tomatoes, and often if they're indeterminate, they could be huge, right? So I say people dig up the root ball, shake off most of the dirt, but we always like to have at least a pint of soil around the roots to keep things moist. And, you know, like Marianne said, put that in a plastic bag, tie it off at the base of the stem, and then just cut the stem off about 12 inches above the ground. So you could put all that in another plastic bag. And then you could take sections of the main stem that are symptomatic maybe, or that have lateral branches with leaves that are symptomatic. And you could put those in a separate bag and package all that so it doesn't get damaged. So there are workarounds. Can you think of anything else, Marianne?
I think that's pretty good. I mean, like Elizabeth said, some samples will bend easily, like, you know, cucurbit samples, you know, cucumber and cantaloupe, and then it's not really a problem. But yeah, I think her recommendations are good on that.
Great, thank you. We had a question on the slide that was the samples for diagnosis of fruit. Do you know what was wrong with the apple in that picture?
Oh gosh, you know, I should have looked up, because there are a couple of things that can look like that. So one thing is called brook spot, and then there's an abiotic problem, and then there's a biotic problem that look very similar. So basically we can't tell them apart unless we culture. And that is an old picture, so I can't remember which one of them it is. Do you know, Elizabeth?
I don't. I saw that question and I thought you would know since it was in your presentation. I thought maybe it could be cork spot or
cork spot or brook spot. So cork spot is the abiotic problem. I think it's like calcium deficiency or boron or something. And then brook spot is a fungal disease. And typically it turns out to be the abiotic problem on that symptom. So we don't see brook spot that much.
Thanks. The next question is about submitting samples. So plant disease samples, so we've had questions about both digital samples and the physical samples. They do need to go through county extension offices, correct? Individuals can't send samples directly to your lab.
That is what we try to encourage, because the mission of our lab is to be a diagnostic service to the extension agents. So if you are a master gardener who's helping out an extension office, and you send it from the office for the agent, then that's one thing. But yes, we are a service to the agents and to the county offices.
Thank you. And then all the information for submitting samples is on the website. We have posted that a few times, so you can read through how to work with your county office to get those samples submitted. All right, the next question we had is, what is the magnification of your ID microscope at the lab?
Yeah, I guess the highest magnification we can go is 400 times with our compound light microscope. And then if we're looking through a stereoscope, I think the highest there is 60x, right, Marianne?
It's 63 on those stereoscopes we have. And we could go up to 1,000 on the compound, but then
that's
a special
true.
lens, and we typically don't use that. We can typically see what we need to see at 400 times the magnification for spores. And then for things like fruiting bodies, which are structures that many fungi produce their spores in, you can see those with the stereoscope even much lower than the high magnification on those scopes. So you can see a lot, just with 10x magnification, like a good hand lens, if you don't have a microscope. A good hand lens is probably better than a bad stereoscope, I would say.
Wonderful. Kind of going off of that, could you describe a little bit more about the culturing process? So the process that takes place in your lab to help diagnose some of those samples.
You want to take that, or you want me to take it, Elizabeth?
I'll let you. You've been teaching the students the culture.
I made a whole video of this process for my students this year because I couldn't show them because of COVID. So how do we culture? Well, if we need to culture, then we take the plant part that needs to be cultured from, and we wash it off so we don't have soil on it, and then we do what's called surface sterilize the tissue. So basically you want to kill off all the microorganisms that are on the surface of that tissue, because there are a lot of microorganisms on things that are not causing the problem. So you kind of want to get rid of those. So we surface sterilize usually with a 10% or 5% solution of bleach, and do that for a couple of minutes. And then we rinse the tissue and blot it dry. And then we do all this in a sterile transfer hood. So it's a sterile environment. And we flame our scalpel. We dip it in ethanol and flame that ethanol off so the scalpel is sterile. And then we cut into the tissue, and we're looking for that junction of the healthy and the affected tissue. So if you didn't send that, we're probably not going to culture. But if you did, then that's exactly where we're going to go. We're going to cut away the surface tissue, take some chunks of tissue from that edge, from inside, like the stem, or if it's a leaf, we'll just do the whole leaf, edge of the lesion on the leaf. And then we put those on different nutritional media. So we have a variety of culture media that we have stocked. So that's another thing our student helpers do, is they make that culture media. And we kind of have to know sort of what we're looking for. So we have some media that are non-selective, that anything will grow on. And we have some other media that are selective. Like if we know what pathogen we're looking for, we can use those media, and that pathogen will grow out and it won't be interfered with any other kinds of organisms. So we'll typically culture on maybe 6 culture plates for one sample. That's why we need enough tissue to do that. And the culture media are they're nutrients that are dissolved in agar, and then we pour the agar into the culture plates and it solidifies. So it's like a gel-like surface. And so we put the plant tissue right on that agar media. We stick those plates in an incubator at 28 degrees centigrade where a lot of pathogens will grow. And then we wait until something grows out from the tissue. And luckily for us, most pathogens will grow out within a couple of days. There are exceptions to that. And sometimes you have to wait for the organism to make certain structures. So it can be sometimes like 10 days for certain pathogens. And so that's what we do. And we have our student helpers culture from this plant tissue. They put the plates in the incubator, and then Elizabeth and I come in, and we've each got a shelf in there, and we'll come in and every morning and go through our culture plates and see what has grown out and what we can answer. So do you have anything to add to that process, Elizabeth?
Well, I was just going to say for bacteria, it's a different approach, but there might be some other questions that we want to answer.
Yeah.
I don't know if there are.
Looks like that covered it. Another question that came in was an EMG wondering what more they can do on their side. So prior to sending in samples. So specifically, where do you see the line being drawn between the work EMGs are doing in their extension offices, including looking at samples under microscopes, and when further work might be needed, say from your lab, like culturing, especially when samples might have multiple problems, which can be more difficult for EMGs to diagnose with what they have on hand at their help desks.
Do you want?
I'll let you answer that,
Okay.
because you've been doing this diagnostics with the master gardener, so you have a better handle on.
Okay, well, first of all, that's a very good question, but it's very case specific. So a lot of times if you're trying to diagnose things visually or with minimal magnification, you may be able to get down to 2 or 3 different possibilities, and you may not know which one is the real answer. So I think if you ever have a question about that, then it would be appropriate to send it to the lab or to send digital images, and maybe we've seen enough of it that we can say right away it's this and not the other thing. So I think it's a matter of how confident you feel, and how confident you feel is going to depend on sort of how much experience you have doing it. And the more you do, the more confidence you get. Like there are certain diseases or plant problems, once you've seen them, you know what it is every time. But I'd say it would relate to your level of confidence. We're always happy to get a sample. And, if it's an easy one, that's great. We can feel good about answering it very quickly. And if it's a more complicated one that we have to culture from, then we'll have the sample. I will say that if you ever suspect that you have a root disease, then you will probably just want to go ahead and send us a sample, because root diseases, the symptoms of root diseases on the plant are kind of, not very specific. Any root problem will cause a plant to wilt or turn yellow or be stunted. And so you're not going to know which type of root disease or root problem it is unless you send us a sample. And basically for most root samples, we have to culture. A lot of plant tissue on the foliage and stems, we can find this like fungal spores, or we can find the bacteria with the microscope. But with root diseases, we typically cannot. With root diseases, we typically have to culture and get it to grow out on the culture plate to recognize the pathogen. And also for nematode samples, you're never going to diagnose a nematode sample unless you send us the soil, because the nematodes are microscopic. Does that answer your question, or do you have any follow-up questions on that?
I think it does, but the Extension Master Gardener who submitted it, please feel free to, it looks like that did answer the question. So thank you for that.
Okay.
And I think that was our last question that came through. So again, thank you both very much for joining us this afternoon. And thanks to all of our Master Gardeners who were able to join us today and for other sessions in this series. I'm posting the webinar archive again right here in the chat box. So all the links for the recordings will be there. And I hope you have all enjoyed and learned as much from all of our specialists and our staff at the labs as I have. So thank you all very much, and we will see you all later.
Thanks, everyone.
Thank you.
Great to have this opportunity. Have
Yeah,
a good day.
thank you.